Sometimes at the end of the assay, what we see that the results are not consistent. The results are quite out of the way that we fail to interpret those results. So इस सूरत हाल में हमें जो है वो वापस जाना पड़ता है और देखना पड़ता है हमें कि where we made a mistake. That we call as troubleshooting. MTTSA करते हुए क्या प्रॉब्लम्स आ सकते हैं और उसकी पॉसिबल रेमेडी क्या है, अभी हम उसके हवाले से थोड़ा सा बात करेंगे. Sometimes we see that MTT reagent is blue-green. Then in that case we should not use that solution. We should discard it and prepare the new one and store it at 4 degrees centigrade in dark, because that change in color may be because of the light effect, that the solution is turning blue to green. That is an indication कि जब आप इसका metabolism करेंगे cell ऐसे में, then it should turn towards the purple color or to change the color. Blanks sometimes gives high absorbance readings. Whenever we set up some experiment, we have some negative controls or we have some blanks. That means in those wells there were no cells at all, only water or only the liquid was added to have a blank value. In that case, we may expect some contamination in our culture conditions. We need to use aseptic methods, aseptic techniques to rule out any kind of contamination. Absorbance readings sometimes they go too high, that sometimes the value comes too high from the range. In that case, what we need to do, we need to decrease the density of the cells at the time of plating. We need to optimize. जहाँ हम बाकी चीज़ें ऑप्टिमाइज़ कर रहे हैं, वहाँ MTTSA के लिए हमें ये चीज़ भी ऑप्टिमाइज़ करनी है कि स्टार्टिंग सेल पॉपुलेशन क्या होनी चाहिए. अगर हमारे पास एक 96 वेल का अपना एक वॉल्यूम, एक एरिया है, उस पर 10,000 सेल्स प्लेट किए जा सकते हैं एक वेल में, या फिर 100,000 सेल्स प्लेट किया जा सकते हैं. वी नीड टू ऑप्टिमाइज दिस नंबर एंड देन वी विल बी इन अ पोजीशन टू गेट रिड ऑफ दिस प्रॉब्लम ऑफ एब्जॉर्बेंस टू हाई. Then we do this test in replicates. 2, 2, 3, 4 replicates are being done at the same time, and each individual replicate is going to have different value. Then that means that your handling has some problem. While pipetting cells in one well, pipetting cell in the other well, pipetting cell in the third well, you did not handle the volume properly, or the pipette which we are using may have some error. The calibration needs to be done. So needs to check the accuracy of the plating, that how good you are in plating, to ensure the equal volume should go in each well, so that the replicates should give the similar, more or less similar values. Absorbance readings comes too low. In this case, we have to look at multiple points. Like we may have to look at the cell density which we plated. We may need to increase the cell density. We may need to increase the time of incubation with the MTTSA assay or with the detergent reagent. We may need to check the culture conditions are appropriate or they are inappropriate, because the culture condition may be in a position to provide the good ambience for the cells to settle down, or the temperature or the humidity or the carbon dioxide availability, they may be the limiting factor. Then increasing the time of culture for plating or cell recovery. Again, sometimes what happens, the cells are not fast growing. They need more time to settle down. We need to look at these factors if we observe any low absorbance values. Now, what is the advantage and disadvantage of MTTSA? The advantage includes that it is inexpensive, a not so costly test. That MTT is metabolized by all cells. The assay can be used with all cell types. A metabolism in any cell type happens, so this assay is applicable to any cell type. Then we may not have to, in fact we have not go to change these cells. We are not required to shift the cells from one plate to the other plate. No cell transfer is involved. That is the major advantage. Everything is being done in one well. There are few disadvantages at the same time. The sensitivity is lower as compared to fluorescent or any luminescence assay, particularly with those cells which do not rapidly proliferate or which have the low metabolic activity. So we are relating MTT assay, its results with the metabolism. If some cells have low metabolic activity, we will not be in a position to have results in a required time span. Then because of ELISA plate reader, the assay is not linear over a broad logarithm cell proliferation range. That means we cannot go for a longer term assay because of the 96 well plate, the space available for cell proliferation. Exposure to MTT and formation of formazan crystal itself is cytotoxic. So this cytotoxicity, although we are not going to use those cells which we have used for MTTS assay, but this formazan crystal may also have some effect, the cytotoxic effect, and may change the morphology of the cells. We cannot take multiple time points in this assay. It is only one step assay that we can take only one observation, one reading can be taken, and then the insoluble reaction product, that is the formation of formazan, that needs to be resolubilized. This is an additional step which involves some acidic and some chemicals which may be toxic if not handled carefully. So these are very few disadvantages as compared to the advantages. So that is inexpensive assay. People in the lab, they prefer to do it because of its advantages over disadvantages.